So here's an example of subliminal backmasking and how you can interpret things a certain way.
Another one bites the dust. Another one bites the dust. Ow! Another one bites the dust. Hey, hey! Another one bites the dust.
So we have this tendency to interpret information. We piece it into something that we understand, a pattern, even if it actually isn't following that specific pattern. We'll interpret it that way. So the argument with subliminal perception is that it's actually not necessarily 100% effective. For the most part, they can't make you do anything that you wouldn't already do. And really what it's just doing is activating concepts in your mind. So it doesn't necessarily lead to behavior. So they did a study, and it's very similar to the one that we just discussed that was more of a fraud, but they tried to see how you could prime people to drink. So they primed people to drink more of a juice. But first what they did is they gave them cookies, and then they gave some people something to drink after the cookies, and then they gave another group nothing to drink after the cookies. So they did this so that one group was thirsty and the other group was not thirsty because they had already had that thirst satiated. And then they gave them another task where all they had to do was answer some anagrams, but the anagrams were about drinking. So they were subliminally priming you to think about drinking or being thirsty. And then after that, they offered them more liquid, so more juice. And what they found is, so you see here, you have people who were thirsty in this one group. So this group of people were not given anything after the cookies. And then this group of people were already given a drink after cookies. And then the black bars are representing people who were given those primes in the anagrams. And the gray bars are representing people who were given an anagram that didn't prime you to want to drink. And you can see that when they were offered a drink, they measured how much of the drink people had. And you see a large increase in those people who were, one, thirsty, and two, they were primed to think about being thirsty. So it had an additive effect where they were more likely to drink more than the other groups alone. So in that sense, you can have effective subliminal perception, but it doesn't work with everyone, and it doesn't necessarily ensure that someone's going to behave a certain way. It just increases their likelihood by activating a concept in the mind. Now moving on to the visual system. Obviously, our visual sense system is how we're perceiving light and sensing the world around us. And it looks like this. So you have your lens back here, and the pupil, which is a hole for the light to go through, the iris around it, and the cornea. Back here is the retina. And you'll notice when an image comes in, it comes through here. The lens will bend the light so that you can see an entire image. So it bends it to focus. And then the image actually ends up upside down in the back of the retina. Now in class we talked about the blind spot. And we were using that exercise with the cat and mouse to show where that occurs. So this is the blind spot right here. And there's a blind spot because your optic nerve is going through there. So there's no cones and rods. Now, when light enters in the back at the fovea, it goes through this neural impulse and you can see the plan. So you got cones, which are rounder, and then rods are long and thin. So there tends to be more cones in the middle, and the rods have a larger clumping in the peripheral. And that's why we're good at dark. In the dark is the rod. So when we can see in the dark, that has to do with the rod understanding of how the information is getting interpreted. Where the cone is for more vivid color, so it's more active in our center peripheral, in the center of our vision. So then it travels through all these different cells. It goes through something called the bipolar cell, then the ganglia, then it travels through the optic nerve to the thalamus, and then from the thalamus it travels to your occipital lobe. And there's huge neurons. So basically it's one giant neuron that's going all the way from your optic nerve to the thalamus, and then one giant neuron going from the thalamus to the back in your occipital lobe. So we've talked about this before in the biology lecture. Remember how when you're feeling things on your left side or your right side, they're actually crossing over to the other side of the brain. Your vision is very similar. When you look to the right, so this is supposed to represent the eye, it crosses over. And then when you look to the left, that crosses over. So the left side of your vision goes to the right side of your brain and vice versa. And you should remember this from when we had the video with the man who had the split brain procedure, right? And he could only recognize things from the left side of his vision on the right side of his brain. So he had problems, right? If it was on the left side, he could draw it, and if it was on the right side, he could verbalize it, but not if that couldn't get switched around. So when this crosses over, this is actually called the optic chiasm, is the crossover point right there. And this is repeating what I said on the last slide. And I just realized I was showing you the image backwards because look, the eyeballs are over here. Haha, those are not eyeballs, which would not make any sense. Eyeballs over here, but it's the same idea, right? It's crossing over with the optic chiasm, which was the point. Oh, I can't read my own slides. Okay, color vision. We talked about this a little bit as well. You have the trichromatic theory, and this is talking about how you have three types of cones. You have a cone that recognizes the red, a cone that recognizes blue, and a cone that recognizes green. And you can combine these cones to form different colors. So that's one theory about how vision works. And obviously this is how you get the other colors. So if you combine red and green, you get that yellow. So the cones are working together. And there's an image of how that's coloring to form different colors. Purple would be here, yellows here, blues or green and blue is a different color. Oops, sorry. But so that's the idea of that. We did this in class. I'm not going to give a lot of time, but if you weren't there, just stare at the black dot and then pause the video and then look at this and you'll see color. But most of you had this in class, so we're going to move on past it. But this is using something called an opposing process. And the problem with the trichromatic theory is when they first developed this theory, it didn't account for this after image that occurs when you're staring at those colors long and then they disappear. So what happens is when you look at green long enough and it disappears, you see red. When you look at yellow, it'll turn blue. Those are how after images work. So once that color disappears, you get a flood of a different color. And this is where you get that opponent process theory. So you're getting opposing colors that occur because the visual neurons get stimulated by one of the colors, and then it gets inhibited from another color. So it's like you just wear out that neuron. And then when it gets worn out, it becomes inhibited. So that's just explaining that with the green-red system. You're looking at green, and then that becomes fatigued. So then the red will rebound. So that's how the after image is working. So both of these theories are correct. It's not one or the other. You have both theories there. So the trichromatic theory is really what's happening as the raw images enter your visual system. And then your opponent process theory is actually occurring afterwards. Oops, sorry. So that's how you're getting the after images, is this wearing down of the sensation. So we talked about this with the color blindness and how there's color deficient vision. So you should be able to see the 6, the 12, the 42, and the 2. And if you can't see it, you might have color blindness. And this has to do with having a defective cone. So usually what's happening is whatever cone is picking up that certain color, your cone is not working properly. So in other words, if your cones aren't correct, so you can't see the orange, and that would be your problem, not being able to distinguish that blue from the orange, because whatever cone causes that differentiation is not functioning normally. So there's typically 2 types of cones working for a colorblind person. The third one is just not working. However, you can have people who, maybe lose more than 1 cone, and therefore they have even more dramatic color vision problems. This is the video I showed in class. I'm not going to show it again, but it has to do with those glasses that allow colorblind people to see the missing colors. Now moving to depth perception. We talked about this a little bit too. This is how you learn to perceive the world in 3 dimensions. So you can estimate distance. And here's some photos that show, trickery as far as our ability to have an illusion when we really can't see the depth. So once depth goes missing, everything looks as if it's on the same axis or the same plane. So hence the sand pail looks gigantic, because you can't tell based on the picture that it's closer. The same cues are not there anymore. Same with the glass. So we use different cues for depth perception. The binocular cues are the type of cues that require both of our eyes, and it's what we talked about in class, where there's almost a mathematical equation going on, where our eyes can see the disparity between the left and the right eye, and it uses the disparity in our vision to recognize how far something is. So because our eyes are inches apart from each other, we're getting that slightly different image. And our eye uses that difference, or our brain uses that difference between the 2 images to figure out the distance. So the more similar the 2 images are, the closer we recognize that object to be. Now, the binocular disparity is that what I just described, where we're talking about the distance or between the 2 eyes and how that equals a difference in our 2 images. Convergence is another cue we use, and that's actually our movement of the eye to look at something. So we'll rotate our eyes when we focus on an object, and the more we rotate our eyes, that is actually a cue to our brain also to tell us how far something is. So when I have to look at something much closer to me, say you want to look at your finger right in front of your nose, you really have to rotate your eyes close together, right? And you can feel even the muscles really straining for you to look at something very close to your nose. Where if you're looking at something very different, your eyes aren't rotating to such an extreme degree, and therefore you can tell it's farther away. So these are both binocular cues because we're using both of our eyes and either the movement or the disparity to tell us the distance. Now we also use monocular cues, and that only requires us to use one eye. Now some of these cues seem really obvious, but they're still informative as far as giving us an idea of the distance. So you have familiar size is one, and that's pretty basic. Like, you know the size that something should be. When you see an apple, you know that it should be a smaller size that fits in your hand. So even if you saw a picture with a giant apple, you would recognize, oh, that apple must be very close to the lens because an apple is a small object. So if a large object appears smaller, we know it's far away, and if a small object appears closer, then